Hi, my name's Dave Miklos. I'm founder of the DNA Learning Center at Cold Spring Harbor Laboratory. Since 2002, the DNA Learning Center has been helping Asian students and teachers learn new techniques in molecular biology. So we were very concerned when we learned of the coronavirus and the hardship that it's causing all of you in China. So we thought we'd do this series of laboratories so you could continue to learn about biology when you're forced to stay at home. For the first experiment, we're going to learn one of the key techniques in molecular biology. Transformation is the ability to move DNA from one place into another organism, and it's absolutely essential to genetically engineering any species or producing any genetically engineered drugs or other products. But in the 1980s, when I first started doing this, it was extremely hard to do and could only be done by the best people in the best laboratories in the country. So I didn't know what to do to bring it to teachers until I talked to my friend, Doug Hanahan. And Doug had a really easy way. And he knew the trick of easily getting DNA into bacteria. He shared that with me. And now you'll learn that same technique today from Aaron McKechnie, one of our instructors here. It's really easy and it's really fun, and it's been done by hundreds of thousands of students around the world. So now you'll be able to do it also.
Good evening, everyone. My name is Erin, and I'm here to teach you the Bacterial Transformation Lab. I'm so excited to work with everyone today. Let's get started. I'm going to bring up my first slide on the board. Okay. So we are going to be working with DNA, and we're going to be working with bacteria. So I'd love to start off by having a little talk about what DNA is and why it's so interesting. If at any point during our lesson you have questions for me, I would love if you tried to type them in. I will do my best to answer some of those questions, and I'll also leave some time at the end so we can talk about them. Let's talk about what DNA is. So I have up here a picture of the DNA molecule, which is found in all living things. So is anybody able to tell me what the letters DNA stand for? I'll write it up here on the board for you. So DNA stands for deoxy, ribo, nucleic acid. This molecule is special, and I'd like somebody to tell me, if they can, where we find DNA. I did say earlier that we find DNA inside all living things. Specifically, we find our DNA inside of our cells. So as humans, we're actually considered animals. So we have animal cells. Inside our cells, well, we have skin cells, we have various types of blood cells, we have nerve cells and bone cells. We have so many different types of cells. When we're thinking about DNA, it's so important for us to understand what the molecule actually looks like. It allows us to envision that DNA inside of our minds so that we can better understand how cells access or use that DNA. So let me show you my model that I have here. Okay, it looks a little bit like what's on that board. So this model here and what I have on the board, they're both in this shape that looks kind of like a spiral staircase. Does anybody know what the name of this shape is? This is called a double helix. And the double helix was discovered in 1953 by two scientists, Dr. James Watson and Dr. Francis Crick. And this discovery of learning what DNA actually looks like is considered one of the most important discoveries in the history of biology. Now, why do you think that might be? Why would we want to know the shape of DNA? Well, just like I said before, knowing the shape of something or being able to see it in your mind allows us to study it and think about it in new ways. So let's talk about the information in DNA and why we might want to study that. So DNA has information. I'd like somebody to try to tell me what the information in DNA is actually for. I'm going to draw some pictures up here on the board for you. So I'm going to do my best to draw that double helix, but it won't be perfect. So here we have our DNA. Inside our DNA, we have sections that carry information. These sections with information or instructions we refer to as genes. Genes are very similar to recipes. These recipes in DNA, let me just shrink that up for you a little bit. These recipes in DNA, ultimately, well, what do you guys think? What are these recipes in DNA actually for? These recipes in DNA have the potential to influence various traits within an organism. And so what I mean by traits, this can mean so many different things. Traits can be what you look like on the outside. Traits can be how your body functions on the inside. And it can even affect behaviors. So I left a space in the middle right here. DNA is actually a set of instructions to make molecules that we call proteins. So let me fill that in right here. Proteins can have different shapes and different functions. Just depends on the protein that's made by the cell. So there are proteins that influence, let's say, what you look like, different foods that you can digest. Proteins actually do things in cells. Sometimes people are making more of a protein than we expect or less of a protein than we expect. And it all relates to how their bodies function. There is actually a way for us to cut out sections of DNA if we want to. We also have the ability to glue sections of DNA together. These techniques that I'm talking about are a form of genetic engineering. Genetic engineering just refers to our ability to manipulate or change DNA in different ways. And one of the really cool things about DNA is that your DNA, although it's not exactly the same as my DNA, well, there are a lot of similarities in our DNA. For one, your DNA is made of the same genetic code that my DNA is. And in fact, all living things, our DNA is made of the same genetic code. And what I mean by that is that technically, your cells could read the information in my DNA, and our cells could read any DNA from any other organism as well. What makes our DNA different from other individuals or other organisms is not actually the code itself, but it is the sequence or the order of the code. You guys may have heard about this. DNA has a set of chemicals, let me show you my model, on the inside of the molecule. So here, let me untwist this for you. When you untwist it, you can see actually that it looks kind of like a ladder, right? All right, so the molecules within the DNA that I'm talking about are right here. These are our base pairs. So our base pairs in the DNA can be adenine, thymine, guanine, and cytosine. Sometimes we just refer to them by their single letters. So you might call them A's, T's, C's, or G's. And your DNA is made up of those A's, T's, C's, and G's, and so is mine. And what I'd like to explain to you is that what we'll be doing today is we're actually going to give living bacteria cells foreign DNA. We're going to give them DNA that's not theirs. It comes from a completely different organism. They should be able to take in that DNA if we do everything correct, and they'll be able to use that DNA, and they should be able to make new proteins. So cool. If everything works, they should show us that they have new traits as a result of the experiment. So, scientists have actually been doing bacterial transformations for many years now. The bacterial transformations that can be of highest interest are those that we use to actually help people who have diabetes. Has anybody ever heard of diabetes before? So diabetes is a genetic disease that has to do with the production of a protein called insulin. So if an individual is not making enough insulin, that protein, what insulin does, is insulin actually helps our bodies to regulate or control glucose levels. So if somebody's not making insulin properly, they're not able to control their glucose levels. So they may, it depends on the individual, they may actually have to take injections of insulin. So where do you get insulin from to give to somebody who has diabetes? A lot of people think, well, maybe we could donate insulin. Could I donate my extra insulin to other people? But that's not really a possibility. So what scientists have come up with is they have come up with cutting out the gene for human insulin from a human cell and sticking it into bacteria cells. So we actually use bacteria cells as little factories to make human proteins for us. It's amazing. Let me talk to you about bacteria on the board for a second. All right, so let me draw you my little bacterium. Bacteria can have different shapes. So this is just a general picture for you. Inside a bacteria cell, they do have their main chromosome. They have their DNA. In addition, they also have ribosomes. Does anyone know why ribosomes are so important? So ribosomes, what they do is ribosomes actually read the information from the DNA and they build or put those proteins together. So really, these bacteria cells, although they don't have as much stuff inside as our cells, they are very successful because they have just what they need to survive. If I were to take, let's say I were to take the gene for human insulin. I'll draw that up here right now. Let's pretend this is from a human cell and it's the gene for insulin. If I were to put that DNA, let me shrink it up, right inside of this bacteria cell here, what I suspect may happen is that bacteria cell would probably destroy that DNA because they don't really know what it's for. Well, if we want to introduce new DNA into bacteria, we have to take advantage of one really cool thing about bacteria. Some bacteria cells have additional DNA other than their main chromosome. And this DNA is called plasmid DNA. Plasmid DNA, okay, is actually circular. So let me draw a circular fragment of DNA right here. I want you to imagine if I had a line of DNA and I took the two ends and stuck them together, I would have a plasmid. I'm going to make this a little bit smaller here, okay? Plasmids can exist inside of bacteria cells, and what bacteria can do is they can actually clone their plasmids, just like that. They can make copies of their plasmids. They can also share their plasmids with other cells. So as scientists, what we do is we take these plasmids. Let's take this plasmid, and we're going to put it right here. Oh, I don't know if it'll let me do this. Let's try. Well, I guess I'll just draw another one. Okay, here's another one. Let's say I have this plasmid. What I can do is I have the ability to cut the DNA open. So I can actually take some little molecular scissors and cut that DNA right open. I can even remove this section here. And I can then take my gene for human insulin and I can glue it in right here. So look at that. I've created my own plasmid using the bacterial's plasmid. I've just modified it or adjusted it in some way. If I do it the right way, the bacteria will not realize that I've messed around with things. So I can then take that plasmid that I've created and I can put it into a living bacteria cell. If I'm successful, the bacteria should read that DNA and they should make that protein coded for on the gene that I inserted. So if I put a gene in for human insulin, that bacteria cell should be making human insulin for us. All we would have to do then is just grow a lot of that bacteria, which is so simple. We can then break those bacteria cells open and isolate the protein. So this is exactly how they make human insulin today. They use bacteria cells. We are not limited to just making human insulin. We can actually make any protein that we want. So today, what I'm going to show you guys how to do is I'm going to show you how we can actually take these plasmids, get them inside of the bacteria cells, and have our bacteria make a jellyfish protein. So we'll be working with jellyfish DNA today. The jellyfish that we work with has an amazing trait. The jellyfish has the ability to glow in the dark. So let me show you on my board. We are going to take some DNA from our jellyfish, and we have it already inserted into a plasmid for us. Let me draw my little jellyfish here for you. So the jellyfish that we're working with, the species is called Aquaria victoria. And this is a jellyfish that can glow. The jellyfish makes many proteins, but the protein of interest for us today is one that we call GFP. GFP stands for green fluorescent protein. And if we studied the jellyfish DNA, we would find that there is actually a gene within their DNA that codes for that protein. So that gene is just specifically for this protein. If a cell makes GFP, that cell should have the ability to glow green. So I have a question for you guys. Okay? Do you think that human cells make GFP? What do you guys think? Well, I can't see inside of my body right now, but I would suspect that at least on the outside, I'm not making any GFP because I'm not glowing. One of the reasons that we love GFP is because it's very easy to see when a cell is making this protein. So naturally, humans don't make GFP. But what do you think? Could we maybe cut out the gene from the jellyfish DNA and insert it into a human cell? I guess technically we could do that, but there are a lot of moral and ethical issues involved with that, about making humans glow. Would any of you guys want to glow? I don't know if I would. So we can actually do this with bacteria, and I want to show you that they've done this with other organisms as well. So let me show you some fun pictures. Over here, I've got a whole bunch of pictures for you. This one right here is actually of a very small worm that we use a microscope to see. This worm right here is actually the first organism that they transferred the GFP gene into. What about this picture here? You see these mice? Well, mice do not naturally glow. So when you're looking at these mice right here that have the ability to glow, those mice have been genetically engineered with the GFP gene. So when we see experiments in multicellular organisms like mice, anything that we do in a mouse, the idea is technically we should be able to apply that information to humans. So if they can make mice glow, technically they could make humans glow. Although I don't expect scientists to be making humans glow anytime soon, the reason that this experiment is so exciting is because, well, it's showing us that it's possible to take a foreign gene, a new gene, and insert it successfully into these mice. That means we could do that with humans too. You don't have to make humans glow, but you could have human cells make new proteins. This is a way of possibly eliminating certain genetic diseases and even just helping people live better lives. Very exciting. What I'm going to show you how to do today is introduce plasmid DNA into these bacteria cells. I'd like to just show you the genes that are present on the plasmid we'll be working with so you understand why everything is important. So let me draw for you our plasmid. All right, so our plasmid, as I've discussed before, is going to have that gene from the jellyfish. So one of the sides of my plasmid, I'm going to draw it over here. One of the sides of this plasmid actually has that jellyfish gene in there, so our GFP gene. There is a second gene that's important for this experiment, and I'm going to put it on the other side for you. I just have to shrink up my plasmid a little bit so I have enough room. Over here, we have a second gene that is for something called ampicillin resistance. Has anybody ever heard of ampicillin before? Ampicillin is what we call an antibiotic. Antibiotics are medicines that can kill or even inhibit or block the growth of bacteria cells. So generally, when we think about how antibiotics work, if I have a whole group of bacteria cells and I add an antibiotic to them, I would expect those bacteria cells to either be killed off or to not be able to grow. So antibiotics are incredibly useful, especially if bacteria are making us sick. This gene is a really special gene because this actually allows our bacteria cells to resist this antibiotic called ampicillin. Now, what do you guys think? What does that mean if bacteria cells can resist an antibiotic? It means that that bacteria has the ability to protect itself from that antibiotic somehow. And there are different mechanisms that bacteria can use to protect themselves. In this case, I want to show you just why this is such an important gene for us. So I'm going to draw another picture of some bacteria for you guys. All right, so let me draw my little bacterium here. We have our DNA again in the center. We have our ribosomes. And what I expect is that in our experiment, we should have a whole bunch of bacteria. So I'll draw a lot of cells out here. Even in a successful experiment, what happens is that the bacteria cells most likely will not all take up the DNA we want them to. And that's perfectly fine. Even if, let's say I just get this one cell to take up the plasmid that I'm interested in, that one cell should be able to make some GFP. Let me draw some GFP. And that cell should be able to glow. The problem here is that these other cells, these non-transformed cells out here, these ones, all these ones, they are going to grow on top of my glowing bacteria, and then I'm not going to be able to see if my experiment was a success. So I have given my glowing cell over here, I've given it the ability to resist the antibiotic ampicillin. So if I apply the antibiotic ampicillin to this environment, the other cells here, what I will expect is that they will not be able to, they won't be able to replicate or grow. So I'm just going to get rid of them. They're not going to be able to grow. But this one that can glow here, this one, because it has that antibiotic resistance gene, it's going to be able to replicate and copy itself. And all of those little copies are going to be able to glow as well. So I'm going to have a whole region of bacteria here that have the ability to glow. We call that region of bacteria that start from a single cell, we call that a colony. So I should have a nice green glowing colony if this all works. Hopefully at the end of our experiment, we should have way more than one glowing colony. I hope to have hundreds. Let's get started on the experiment. What I'd like to do is actually show you some of the instruments that we use for measurement in this lab. This is called a micropipetter. We will use different micropipetters in the lab, and they work exactly the same way. The only thing that's different about them, other than the color on top, is that they measure different amounts of liquid. So they measure in the same type of liquid, in the same unit. Oh, that's our cat. I wasn't going to show you guys that yet, but that's a transformed cat glowing in the dark. Let's come back to our pipettes. So our pipettes measure in the unit microliters, which looks like that. There are 1,000 microliters in 1 milliliter. At the start of our lab, we will be measuring 250 microliters of a solution called calcium chloride. Before I actually measure anything out, I'm going to label some tubes. In this experiment, I will have 2 tubes. So I will label 1 tube with a plus and 1 tube with a minus. Our plus tube will be our experiment tube. That will have bacteria and plasmid DNA and some calcium chloride. Our minus tube over here, this one over here, is actually not going to have any plasmid. So this is going to act as our control. Let me show you the tubes that we're going to use, and I'll show you just how simply I label them. So I'm going to be using these 1.5 milliliter tubes right here. Okay, I have 2 of them set for our experiment. And throughout my work, I want to keep everything labeled, easy to identify, and sterile. Does anybody know what sterile means? Sterile refers to being completely clean or free from contamination. So I want to keep everything clean and separate. Let me label my tubes right now. I'll show you. So I put a plus on that tube, and I put a minus on that tube. Just like that. Very simple. Okay. I'm going to measure out now my 250 microliters of the calcium chloride liquid. So I'll be using the blue pipette because it measures the larger volumes. Our pipette happens to be set on 250 because I was just using this pipette yesterday. But if at any point you need to adjust the settings on these instruments, they're so easy to use. There's a dial at the top. You can change that, and it actually, I don't know if, oh, you guys can see that. It changes the numbers. So for me, I'm going to set mine at 250 microliters. Remember, we want to keep everything sterile, so I'll be using this box of plastic tips, and I'll be changing them whenever necessary. So let me place that down there, put a tip on, and I'm going to pick up my tube of calcium chloride. Opening my tube, I hold everything up to eye level, and I'm going to pipette slowly. Placing the pipette in the liquid, I'm going to slowly let my thumb come up. I'm going to pick up either tube. I can start with either one. And I'll be adding the 250 microliters of calcium chloride to that tube. At this point, I'm going to place my tube on ice, and I'll explain to you shortly why the ice is so important. I'm going to remove another 250 microliters. Slowly pipette. I can discard my extra. And I'm holding up my new tube, slowly adding, closing that up, and placing that on the ice as well. At this point, I'm just going to eject the tip and move on to the next step. I think you guys are going to find this step pretty exciting. I'm going to transfer some bacteria off of a petri dish. Let me show you. So let me show you an empty petri dish first. So this is a petri dish. I don't know if you guys have ever worked with these or seen these before, but this petri dish, it just has food for bacteria in it, that's all. Look at this dish. Let's compare. There is also writing on this dish over here, but I want you to see all that smeariness. You see that kind of beige colored material that's swiped or painted all over the plate? Those are the bacteria cells. So I'm going to do my best to sterilely transfer the bacteria that's on the dish into those 2 tubes that I just labeled and added calcium chloride to. In order to do this sterilely, I'm going to use some flame. So let me pull my hair back so that everything is safe. And I've got this great little burner here, which I'm going to turn on. It's just a gas canister, and I'm going to apply a spark. So let's turn the gas on. There it is. And there we go. All right, I do like to work, oh, let's try that again. There we go. I do like to work close to the flame to keep everything as clean as possible. I really consider this area right around the flame a zone of sterility. The heat is going to push anything up that might be falling into my experiment. I don't have to work so close to the flame that I'm going to get burned. I would never want to do that. But I do work nice and close so that everything is clean. This is an inoculating loop. Can you guys see that circle? Maybe I hold it like this. I don't know. Let's see. Oh, there it is. I can see it. It's a little tiny circle. What we do with the inoculating loop is we're actually going to try to scrape or lift the bacteria off the petri dish. I don't want to take any of the actual gel that's from the dish, so sometimes this can be a little tricky. First thing I'm going to do is I'm going to sterilize my inoculating loop. Now keep in mind, this is a piece of metal that's basically on fire. If I take this flaming hot piece of metal and I touch my living bacteria cells, what do you think might happen to them? They're probably going to die. So I have a little trick here. I'm going to sterilize my loop, and just for a moment, I'm going to press that loop into the gel to cool it off. At that point, I'm going to use my loop to kind of scrape and lift the bacteria out of the dish. The bacteria sometimes are a little bit slippery, and this can be a little bit frustrating. I think I'm doing a good job, though. I'm going to go with that. I don't know if you guys can see this amount here. It may not look like a lot, but that's actually a lot of bacteria. So I'm going to pick up one of my tubes with the calcium chloride. Doesn't matter which one, they're both going to get the bacteria, and I'm going to place the bacteria into the tube. I'm going to do my best now to kind of swirl off the bacteria into that liquid. I think I did a good job. Let me show that to you. So that looks nice and cloudy. That's what I want. That's an indication to me that there are a lot of cells in there. So at this point, I'm going to place this back in the ice. I want to do exactly the same thing for the other tube. So I'm just going to sterilize my loop. Looks good. Don't forget to cool it off. And then I'm going to scrape and lift some bacteria again. I think I got a good amount. Let me find my other tube. There it is. Open that tube, place the bacteria in, and swirl it off. Okay. I'm going to sterilize my loop one more time, just to keep everything clean in the lab. At this point, I'm going to turn the flame off and push that out of my way for a minute. Okay. Now, I don't know if you guys can see this, but there are actually a few clumps of the bacteria right in the tube. And I do prefer if it's more evenly mixed. Oh yeah, right on the bottom. Can you guys see that little white clump down there? What I'd like to do is I want to break up that clump. So I'm actually going to use the pipette to do that. So I'll get myself a new tip. I'm going to open my tube, hold everything up to eye level, and I'm going to just very simply lift the liquid out and replace it. At no point am I actually going to remove the liquid from the tube. I'm really just using the pipette as a method for mixing We call this step resuspending the cells. That looks pretty good. I'm going to change my tip and get myself a new one, and I'll be working with the next tube. There we go. Looks pretty well mixed already, but I just want to make sure everything is broken up. All right, great job. Now, I'm going to add the plasmid DNA to one of the tubes. Do you guys remember which tube we said was the experiment tube and which was the control? Actually, I wrote it on the board, so hopefully you guys can figure that out. This one is our experiment tube, so I'm going to add the plasmid DNA just to that tube. In fact, I want to make a list. I want to keep track of what I'm adding to the different tubes because it's very easy to get confused. So whenever I do my lab work, I like to write my information down. So let's start with this. I've got my plus tube. I've got my minus tube. All right, so at this point, they actually have the same stuff inside. So they both have 250 microliters of calcium chloride. And they both have the bacteria. The type of bacteria that we're working with today is a type of harmless E. coli called MM294. We like working with this bacteria, and we like that they're safe. We would never want to work with bacteria that could get us sick, and these bacteria cells are great for transformations. One thing I have to add to the board is what we're about to do. I am about to add the plasmid to the plus tube. I'm going to add 10 microliters of the plasmid, and the plasmid's name is PGFP. Let me show you that plasmid again, just as a reminder. So here it is. This is our plasmid. And in the center, I'm just going to label it PGFP. So as I add this liquid in the next step, which will look to you and to me just like water, I want you to envision again that this plasmid, thousands of them are in that liquid. We are going to add those plasmids into the tube with our bacteria. So I am going to use the gray pipette because I'm only measuring 10 microliters. I'm going to check that my pet is set, and it is. It's on 10 microliters. And now let me just organize. Got my plus tube right there, and over here I have my plasmid DNA. So every time I give these tubes out to my students, they say, Erin, you gave us an empty tube, we need a new one. But I want you to check that out. I want you to see the volume at the bottom. Oh, let me hold it up to my shirt. Maybe that's a little bit better. Is that good? It's still pretty small, right? It's still pretty small, right there. I promise you, there's at least 10 microliters of liquid in there. So I'm going to take my pipette, get a new tip, and draw up that liquid. Yep, there's actually a lot of extra DNA in that tube. Now, am I putting this in my plus tube or my minus tube? Do you guys remember? I'm going to find my plus tube, open that up, and when I do this step, it's important that I place the tip directly into the liquid and then push the plasmid in. I always want to make sure that when I add the plasmid DNA, it's in direct contact with those cells. Put that back in the ice, and I'm going to change my tip. At this point, I like to let the bacteria cells get nice and cold. So I would let these cells sit in ice for about 15 minutes. What I'm hoping to do in the next step is I want to actually push the DNA into the cells. I'm going to do a quick drawing for you, and then an animation, and then we'll do the other steps. Okay, so let me draw a little bacterium for you. So many of the cells that we work with, I would expect that they would have these openings in their cell membrane, which we call adhesion zones. So now let's say I've got a whole bunch of these cells. Oh, hold on. let's say I've got a whole bunch of these cells in there. If I'm talking about the plus tube, I've also added, I guess let's make it in red here, I've also added some plasmids. Okay. So right now, the plasmids are on the outside of the cells. And where I want the plasmids to be is I want them to go into the cells. In order to do that, I'm actually going to take advantage of temperature and how temperature impacts the movement of molecules. Let me show you a cool animation. Okay. So this step is called our heat shock step. This is a wonderful little bacterium, and they have a much better picture here of these beautiful adhesion zones in the membrane. I'm going to zoom in on one of those adhesion zones. So this itself is the adhesion zone right there. Let me see if I can draw on here. So here's our adhesion zone. Oh, it won't let me. It's right in the center. And you'll notice right outside that, in the membrane, we have these very fast-moving phospholipid molecules. And they do carry a negative charge. So what I'd like to do is I would like to transfer, let's say there is a plasmid out here, I would like to transfer that plasmid right through that adhesion zone into the cell. But there are a few issues here. So if I tried to do that, that was our plasmid. Did you see it bounce right out? Okay. One of the reasons that this is an issue for us is that plasmids are DNA, therefore they are negatively charged. And you'll see that we have these phospholipids bouncing around, also negatively charged. So why is that a problem? If we have these two negatively charged molecules and we're trying to force the DNA through, unfortunately, with these two charges that are negative, the DNA is going to be repelled or bounce right back out just like we saw. So the way that we handle this situation is by using our calcium chloride solution. I also like to cool the cells down, and you'll notice that when we do that, let's drop that temperature, that all the molecules slow down. And we can have a neutral environment there. If I use temperature to slow the molecules down, I can also do the reverse. If everything is neutral at this point and everything is slow, I want you to think about what would happen if I suddenly warmed up the tubes really quick, made them nice and hot. What I suspect will happen is that because of the movement of the molecules, we should have at least some of the plasmids move into the cell. Okay, so we're going to warm them up, and hopefully we get a transfer of the plasmids into those cells. At the end of this, I would also like to put the cells in ice to encourage the plasmids to stay where they are. So let me show you this step. So I have my two tubes still on ice. You guys remember? So I've still got my plus and minus tube in the ice. When I do the heat shock step, it's important that I go right over to my water bath, which is at 42 degrees Celsius, and put my tubes directly from the ice into that water bath. In fact, what I'd like to do, let me write this on the board for you. I just want to make sure everything's nice and clear. So we have our heat shock, which is really very simple. 3 differences in temperature. We're starting out on ice, okay, which is 0 degrees Celsius, and I like to do that for 15 minutes. The next step is warming everything up, and we find that 42 degrees Celsius works best, and we do that for 90 seconds. At the end of that 90 seconds, if you remember, I'm going to place the tubes back in the ice. I do that for at least 1 minute in the ice at the end. So let me show you this step. Okay, so here's our lovely little water bath over here. I have it set at 42 degrees Celsius. I'm ready to go. And again, I want to emphasize, we're going to stay in the ice or we're going to be right in here. So when I'm ready, I have my timer set. I'll be taking my tubes, they go directly into this water bath, and they'll sit there for 90 seconds. At the end of that 90 seconds, what I'm going to do is very simply transfer those tubes back into this ice. So we'll pretend that we did 90 seconds there, okay? I'm gonna let my tubes sit in here for at least a minute. So that means I could let them sit longer. During that time, what I usually like to do is I usually like to label my petri dishes. I'm not going to take my tubes out of ice quite yet because I still want them to sit. But if I hold the tubes up to you, do you expect that they would be glowing just yet? I do not think that they would be, for a couple of reasons. So under ideal conditions, so meaning everything's perfect, I would think that bacteria cells have the ability to make a new protein in maybe as little as 20 minutes. But even if they were making that protein in 20 minutes, I'm not going to be able to see it because the individual bacteria cells are just too small. I need to take those cells and give them an opportunity to grow. So one of the ways we can do that is by plating them on petri dishes. So I showed you the petri dishes before. I actually have a set of dishes here, all labeled for us to work with. Okay, so here's one of my dishes. We'll actually be using 4 dishes for this experiment, and most of the information on the dishes is the same. Let me just give you a quick look at what I put on the dishes. There we go. There it is. Okay. So here you'll see I have 4 different petri dishes, and in fact, most of the information on the dishes is the same. So I did draw my initials on the dish, on each dish. There we go. I also put the date, which is important. I want to know when I did this experiment. And up here you'll notice that I wrote PGFP on each dish. What is different though on these dishes is that some dishes have a minus PGFP and some dishes have a plus PGFP. And that becomes important when we interpret the results. You'll also notice that I have some other information here on the dishes. Some of the dishes say LB and some of them say AMP. Let me explain what that's all about. So in our lab, we have so many different types of petri dishes. Some of our petri dishes just have food for bacteria, and some also have antibiotics in them. So the petri dishes that say LB on them, those are just petri dishes with food. The petri dishes that say AMP on them, those actually have the antibiotic ampicillin in them. So I want you to imagine, if I take my bacteria and I plate them onto these dishes, some of the bacteria have added plasmids, some don't. Some of the plates have just food, some have antibiotics. So I want to consider what our possible results would be. Let me erase these little lines here just to make sure everything's nice and clear. All right, so if right here, and I'm just going to circle this just to emphasize, you can see this is a minus plate right here. Okay, that's a minus plate. So if I have an LB plate right here, this is the plate I'm talking about, okay? I have an LB plate, and I'm just adding bacteria from my minus tube. Well, that bacteria is just regular bacteria. No plasmid with DNA was added to that. So if you put regular bacteria onto a dish with food, what do you expect to happen? I would expect that they're probably just going to grow. So what I'll draw is a pattern of growth. Let's see if I can do this. A pattern of growth, oh, one more time, up. So let's try 1. All right, let's try this again. We'll put this down. 1. There we go. Okay, it doesn't want to do it. We'll just do it without. Let's try this. Okay, we'll do it like that. All right, so I expect the bacteria to grow all over the dish in a pattern of growth that we consider a lawn, or we call a lawn. So there's bacteria all over that dish. Now, let's imagine I take this same bacteria that's in the minus tube, and what I want to do is I want to add it to this ampicillin plate down here. Okay? I'm going to add just regular bacteria because we have that minus, right? Remember that? So I'm just adding regular bacteria to an ampicillin plate. What do you think happens if you put regular bacteria on a petri dish with an antibiotic? If you said that you think that the bacteria would not grow, I would agree. So we will not see growth on that plate. That's actually a very important plate. No growth on this plate indicates to us that the bacteria are inhibited by this antibiotic. They're not able to grow in it. Let's consider our plus bacteria now over here. So this is plus bacteria, which means that those bacteria cells had the plasmids added to their tubes. If I did everything and everything worked properly and the plasmids went in the cells and those cells are making that GFP, I should get some glowing bacteria. But I have a whole bunch of other bacteria in there that are just not glowing. Let's try it this way. There we go. So I won't be able to see my glowing bacteria on this dish because everything can grow on there. Let's take this same plus bacteria from that same tube, okay? So we're going to add it to this plate. I'm going to expect we should get some glowing bacteria. I am going to be putting that same regular bacteria, oh no, putting that same regular bacteria that was on this plate. They're all together, they're all mixed. So when I add a little bit of everything, I technically am getting it all over the plate. This plate though, right here, this plate has the antibiotic ampicillin in it though. So the regular cells here, these regular cells, I would expect that the ampicillin is going to block their growth. They're not going to be able to survive on that plate. The only cells that we'll end up seeing on that plate should be our amazing green bacteria. They should be our successfully transformed bacteria cells. So let me show you just very quickly how we transfer the bacteria from the tubes onto the plates, and then I'll show you our results. So I personally, there are a lot of labels on everything, so I like to organize my plates. I take a quick look. These are both my plus plates. These are both my minus plates. Oh, I'm realizing I forgot to add one step. I always like to add a little bit of extra food to my bacterial tubes before I plate them. So right here, I have what's called LB. So this is basically just like a regular petri dish, all the nutrients without any of the gel. I'm going to place 250 microliters of this liquid into each the plus and the minus tube. So let me drop that liquid, nice and slow. I'll pick up either tube, close that up. I'm going to change my tip, keep everything clean. I don't want to mix or contaminate my samples. There we go. Throw that out. Here's my minus tube. All right. So at this point, I actually don't need the ice anymore. I've just placed my tubes right in the rack, and I'm ready to plate them onto my petri dishes. One thing I like to do before I add the bacteria, and you could do this before or after you add the bacteria, is I like to add these little sterile beads that we have that we use as a mechanism for spreading the bacteria across the surface of the plate. So here I've just got a tube of beads. I'm going to just lift open the plate, dump the beads on, try not to touch anything, and close. I will add beads to each plate and try to do so in a sterile manner as best I can. One more. If any of these beads happen to bounce out onto the table as I was doing that, that would be okay, but I wouldn't want to add them back into the plate because they wouldn't be sterile. At this point, I'm ready to add my bacteria, so I'm going to add 100 microliters of cells to each plate. I'll set my pipette now. I just want to be sure that I add the correct bacteria to the correct plates. So again, one trick I use for doing that is just separating my plates out. I'm using the yellow pipette, gonna get a new tip, and let's take, this is my minus, so I'm gonna add it directly to my minus plates. I have plenty of liquid for both plates here. I'll be drawing up 100, lifting the cover just for a moment, releasing the liquid on the plate, and getting another 100 from that same tube. At this point, I can throw out that tube, get a new tip, and I'm going to repeat the process for the plus tube. I'll drop 100 microliters, add it to the plate, draw up another 100, I can discard that, add this to this plate. All right, now all I have to do, this is the fun part, I stack my plates in the same direction and I shake. Do you guys hear that clinking noise? So that clinking noise is the beads bouncing all over the plate. What I expect is that the beads are moving the bacteria evenly across the surface. If I do this for about a minute, we should have bacteria everywhere. At this point, I would take the beads out of the plate, and I'll just do one so you see how I do this. I kind of knock them toward the bottom, hold both sides of the dish, and knock them out. I usually check so that there is no sound and I know they're out. Let me actually show you what the real plates look like, because I did a set yesterday for you guys. The GFP protein, it glows green, but it glows super green when it's under a UV light. So I'm actually going to turn the lights down for a second, turn on our UV light, and show you the special plates. You guys still see me? All right. It's a little bright in here because it's morning over here. Okay, but check this out. Wait, let me turn the light off for a second. All right, I know you can't see very clearly, but you don't see anything glowing, right? We don't see anything glowing there? All right, now let's turn that light on. You guys see that? Let me hold it up light. Whoa! Doesn't that look amazing? Do you guys see that? So these are cells that did not have the ability to glow yesterday, right? We were able to take these cells, here's another plate. We were able to take these cells, give them some jellyfish DNA, and now they have 2 new traits actually. Not only do these cells have the ability to glow, but they actually already have the ability to also survive in the antibiotic ampicillin and grow. So cool. All right, so I just want to tell you guys what an absolutely fantastic time I had working with you. And I'd love to know if you guys would be willing to maybe do a quick survey and tell me a little bit about yourselves. I'd love to know a few details about you, like where you guys are coming from or where you're watching. So let me ask you a few questions, and if you guys would be willing to tell me the answers, I'd just love to know. So first, let's start. In which school are you currently enrolled? You guys should see a whole bunch of answers there, and hopefully, no, yeah? Okay. I do see a pretty cool question up there as you guys are doing this. Won't the UV light kill the bacteria? Oh, that's a great question. So yes, I guess if I left the bacteria on there for an extended amount of time, you are 100% right that too much UV light would kill those bacteria cells. But if you notice, I just did it very quickly. And I can do that, you know, more than once. There's no problem with that. And I could even, now that I have these transformed cells, I could actually take these cells off the petri dish and grow them on another petri dish and make more glowing bacteria. So fantastic question. Oh, you guys have a pen and a UV light. That's awesome. Great stuff. Those are cells. Yes, those are cells. So all that green material that you saw, that was just a lot of glowing cells. Each of those little spots is what we call a colony, which, if you remember, is where we had one individual transformed cell. So one little cell that took up the plasmid and divided and divided and divided and produced that larger area of that glowing bacteria. Oh, you guys are, oh, great. I see you guys answering so many. Great. Oh, how long can it be kept? Do you mean the bacteria itself? So for the back, two things. So one person asked about incubating them overnight. So you can definitely put the bacteria in an incubator at 37 degrees Celsius. And what you would get is, if I do the experiment right now and I put my cells in an incubator tonight, when I come in tomorrow, I should be able to see my cells, because at 37 degrees Celsius, the cells are going to grow rapidly. If I don't have an incubator, I can just leave my cells on the counter at room temperature. They will grow more slowly. So rather than seeing them in one day, I would see them probably in 2 days. I did see someone ask about whether or not you could keep the bacteria. We do actually keep our plates sometimes if they look really good. We might put a piece of what we call parafilm around them and put them in the refrigerator. Technically, you could keep them outside the refrigerator, but what would happen is everything would get really dried up pretty soon. Okay. So could you transfer the plasmid gene to plants or other organisms, or maybe through the zygote? Okay, so all interesting questions. Bacteria makes it really easy to work with bacteria, because as you can see, we can do some very simple steps in a very short amount of time. The more complex the organism may require different steps in order to get those genes into those cells, but you can absolutely get this gene into other organisms as well. So you guys remember the bacteria, you guys know about the mice, and you did see the cat that I showed you, right? You saw the cat. Okay? I want to, if I have a few minutes, oh, I'm going to ask you another question. There it is. Okay? They're okay? You guys are answering? You guys are so good. Thank you so much. So in case you weren't, would you like to come to the DNALC to study more in person? I hope so. I would like to tell you guys that this has been such an incredible opportunity to be able to talk to you guys, being in New York and you guys are in China, but it's like we're together. It's amazing. I would absolutely love to have an opportunity to meet some of you, to work together with you. I have colleagues, I have people that teach with me, and they are amazing, and you guys would have an incredible experience if you came to work and do our labs with us. Okay? I did see somebody wrote about pigs on there. Yep, they do GFP with pigs as well. So again, when we're inserting the gene, I see that about mammals, it's just a little more complicated. Okay, you guys did such a great job. Do we have any more questions? All right, thank you everybody.